Hi everyone. In this slide we're looking at a human blood smear. Now this region here I've actually selected because it has a lot of leukocytes in it. You could scan to other parts and you probably wouldn't quite find quite as many. In this case here we just have a nice region with lots and lots of leukocytes to try to focus on. So we're going to focus in this region. As you can see, even at this magnification, you can see that there's lots of red blood cells here. These are the main component of blood, or at least the formed elements of blood. And so the leukocytes, the white blood cells, are a very, very minor component here. About 2% of all of this formed elements of blood are going to be leukocytes and platelets. Okay, so what we're going to do is we're going to try to find a few good examples of the different leukocytes that we've talked about in class. So let's zoom in. So I'm starting out right now at 100x magnification. 40x you really couldn't make out much detail. So we're at 100x right now, and we're going to move up to 400x to see a little bit more detail. And so what do we have here? So I guess first things first, we have a cell right here at the pointer. Okay, this cell has very clearly 4 lobules at least. So we're looking at a neutrophil, a mature neutrophil. Okay, there's a few more on this slide, on this region here. There's one more here. There's another neutrophil here. Again, another one just above here as well. Plenty more. So again, as you can see, lots of neutrophils on this slide. And again, that makes sense since we talked about in lecture. The neutrophils are the most common white blood cells. Again, this is your first line of defense. So these are going to be plentiful, okay, because they need to be able to be deployed on short notice. They need to be able to enter tissues whenever there's any kind of infection detected. Okay, now this cell over here looks a little bit different. Okay, the cytoplasm is a bit more intensely staining in this case. So let's zoom in on this one just so we can see if we can get something more out of the 1000x magnification. And here we go. Okay, so again, unfortunately I forgot to bring the oil for the oil lens, so I'm just stuck with getting a less than perfect image here. But if you compare these two leukocytes that you see here, the one at the top definitely has a bilobe nucleus, and the cytoplasm is definitely filled with something a bit more intensely staining and is relatively eosinophilic. So this is actually an eosinophil that we're seeing here. Let's zoom out to the 400x. I think we're getting a bit more of a sharp image this way. Okay, so again, there's that neutrophil we were just looking at, and there's the eosinophil for comparison. Okay, so very clearly we have a bilobe nucleus visible even at this magnification. Okay, now if we look at this cell here, whoops, where are you? Here we go. This is a monocyte. Okay, so you can see that this is a larger cell than the others. Also, the nucleus is much larger than the others. The nucleus is kidney bean shaped. Let's try to zoom in on this a little bit more. See if we can get a little bit more detail out of this image here. Okay, so here we can see very clearly again a very large, fairly pale staining in comparison to the others, fairly pale staining nucleus. Again, this is a cell that has to be able to differentiate into several other different cell types, and so it has a lot more active genes, a lot more euchromatin available, and so it's going to have a much more decondensed chromatin, and so larger nucleus and more poorly staining. Again, this nucleus looks somewhat kidney bean shaped. Okay, and the cytoplasm there is relatively basophilic. Okay, so again, a typical monocyte in this case. Let's go go back out again to 400x. Okay, so again, here's that monocyte we were just looking at. Okay, the eosinophil is just up here. Lots and lots of neutrophils visible. That's probably another monocyte here. Somewhat stretched out, possibly a bit of damage there. Okay. Again, lots and lots of neutrophils. There's another one that's more of a band nucleus here. It's probably more of a younger neutrophil in this case. Okay, so we got two cells here that are a bit more intensely staining nuclei. Well, the nuclei are round. Okay, so in this case here, what we're looking at is a couple of lymphocytes. Okay, so let's again try to take a closer look at high magnification. Okay, and so again, you can see here, two cells, nuclei are fairly intensely staining. These nuclei are about the same size as the nucleus, the nuclei are about the same size as a red blood cell, not the nucleus of a red blood cell, that doesn't really exist at this point. But the nuclei of the lymphocytes are about the same size as a red blood cell. And there's a slight bit of cytoplasm visible just at the edge of this cell here, for example. So there's a bit of cytoplasm visible right there. Okay, so somewhat basophilic cytoplasm in this case as well. Okay, let me see if I can sharpen this up a little bit with more contrast. Okay, so here you can see it a little bit better, hopefully. And again, the one below that also has a small amount of cytoplasm visible around that nucleus. Okay, so we have a little bit of cytoplasm. These are both small lymphocytes. Now, by the way, periodically you'll find some of these little clusters, these somewhat basophilic areas or spots there. Those are platelets. Okay, so you can see a few of them scattered throughout. Okay, so what you're seeing here are platelets again. Okay, let me zoom back out again. Let's get back into focus. And again, just scan around, okay? Again, in here, it's kind of difficult to tell, but this is likely to be an eosinophil just based on the fact that the cytoplasm is very, very eosinophilic here. Over here, this one is a monocyte, very clearly a kidney bean or horseshoe-shaped nucleus, a very large cell and a basophilic cytoplasm. Okay, let's move around some more. I already saw these. Again, more neutrophils. Another monocyte. More neutrophils on this slide. Okay. Again, lots of neutrophils. And again, here's a lymphocyte. Here. There's another lymphocyte down here. Another lymphocyte right next to the neutrophil, and a monocyte as well. Again, you can see that basophilic cytoplasm very clearly. Okay. So, here's another nice large monocyte. Okay, right there. Very pale, staining nucleus. A little bit of cytoplasm visible right next to a lymphocyte, so you can see the difference in the staining of that nucleus very clearly. Let's see if I can zoom in on this for you guys, just so you can see the differences more clearly. Okay, here we go. So again, we have a neutrophil, a monocyte, and a lymphocyte right next to one another. And again, very different appearances to them. Again, you might say, well, this monocyte doesn't seem to have a horseshoe-shaped nucleus or anything like that. Keep in mind, you are looking at a three-dimensional cell in 2 dimensions, so you don't always get that perfect view. Again, textbook images are in textbook because they are the perfect example of something. It doesn't always look that way under the microscope in real life. not every single time. So just be aware of that. Don't expect to see the perfect image every single time on your slides. Again, more lymphocyte and more neutrophils. And again, that makes sense because those are the more common cell types. And again, here's another eosinophil showing up in here. Again, a bilobe nucleus and an eosinophilic cytoplasm. It doesn't look very granular, unfortunately, but again, it's just the level of staining, and unfortunately, I can't go to higher magnification to give you a lot more detail from this one. Okay. So hopefully this has been helpful, and we'll see you in the next video.